Later on, the extensions of astrocytes are wrapped around brain capillaries, contributing to the blood-brain barrier. Blood-brain barrier is a physiological barrier that is important to prevent harmful waste products from reaching the brain tissue through blood. Astrocytes are the largest and most abundant type of glial cells, having multiple projections extending from the cell body, which interact with the neurons, blood vessels, and the connective tissue wrapping the CNS mater. Astrocytes control the concentration of Figure 7.32 glial cells of CNS and PNS. The CNS has supporting glial cells as astrocytes, oligodendrocytes, microglia, and ependymal cells which support the neurons of the CNS in numerous ways. Potassium ions which regulate the concentration of neurotransmitters around the synapses. Oligodendrocytes. Oligo, meaning few, are one of the neuroglia responsible for the formation of myelin sheaths around axons. These specialized cells are found only in the brain and spinal cord, the central nervous system. Myelinated axons conduct nerve impulses faster than non-myelinated nerves and provide insulation. Few processes extend from the cell body and they provide support to neurons extending within the brain. Microglia. Another type of neuroglia are microglia, which are the smallest glial cells with short twisted processes extending from the soma. These cells are constantly moving and referred as the phagocytes of the central nervous system. They phagocytize cellular debris, damage cell, pathogens, and involved in immune response, thereby protecting the brain cells. Microglia are the protector of brain as they initiate a response to remove toxic substance upon encountering any. Ependymal cells. Ependymal cells are ciliated and line the fluid-filled ventricles of brain in the central canal, canalis centralis, of the spinal cord. These cells produce cerebrospinal fluid, CSF, in choroid plexus and are involved in the circulation of CSF. Satellite cells. Satellite cells, also known as ganglionic gliocytes. They surround the neuron cell body and ganglia of peripheral nervous system and provide support. They form complete layer around the cell bodies of neurons and ganglia and aid in the maintenance and regulation of chemical environment around the cell body, provide insulation and pathway for metabolic exchange. Schwann cells. Schwann cells are specialized types of glial cells that are responsible for synthesis of myelin sheath around axon and dendrites in the peripheral nervous system. These cells wrap in a spiral manner around a small part of axon. Myelination. Axons and dendrites of a peripheral nervous system are wrapped in Schwann cells. Schwann cells surround the processes of neurons in several layers of membrane called as myelin sheath. Myelin is a phospholipid with whitish appearance and is made up of concentric layers of oligodendrocytes plasma membrane, which aids in faster transmission of electrical impulses. Myelin sheath acts as a layer of insulation for transmission of electrical impulses. Axon is broken at various points, thus forming internodal segments of myelin sheath. Axons with myelin sheath around them are referred as myelinated and those without myelin sheath are called as unmyelinated. Myelin sheath enables faster conduction of nerve impulse. In addition to oligodendrocytes, Schwann also produce myelin sheath. The oligodendrocytes produce myelin sheaths in CNS and Schwann cells produce myelin sheaths in PNS. Space or segment between adjacent Schwann cells of the myelin sheath are known as nodes of Ranvier neurofibril nodes. These nodes depolarize when an electrical signal is transmitted to the neuron cell. The Schwann cells wrap around the axon many times in a spiral manner with Schwann cell cytoplasm and nucleus forming the outermost layer, neurolemma or sheath of Schwann, and multiple layers of Schwann cell membrane lying beneath. The neurolemma is only found around axons of PNS. The interruptions in the myelin sheath are called as nodes of Ranvier. One axon segment between two nodes is wrapped by one Schwann cell. The amount of myelin increases from birth to maturity. The myelin increases the speed of conduction of nerve impulse along the length of axon. The speed of nerve impulse conduction depends on the factors such as diameter of axon, presence of myelin, and temperature. Increase in temperature, presence of myelin sheath, and larger axonal diameter increases the speed of nerve impulse conduction. The action potential. Sudden, fast, and propagating change in the resting membrane potential is known as action potential. Neurons have the property of excitability and thus are capable of generating action potential which helps in communication with target cells. Action potential is caused when a stimulus changes the resting membrane potential. The plasma membrane of nerve cells is made up of a bilayer of phospholipids and has a hydrophobic core, thus only hydrophobic substances can traverse across the membrane without any aid. However, charged particles, hydrophilic substances require the assistance of channel proteins or membrane proteins to pass through the membrane. The movement of ions by various passive transport channels and active transport pumps leads to the generation of action potential. Ions concentration inside a nerve cell is established and maintained by ATP-dependent pumps. These pumps exchange internal NAD+ for external K+, thereby concentrating NAD+ outside and K+ inside the neuron. Sodium-potassium pump needs energy in the form of adenosine triphosphate (ATP), thus it is often referred as at-pace pump. Figure 7.33. High concentration of NAD+ on the exterior indicates a concentration gradient, and the electrical gradient is reduced as a result. Movement of positively charged ions into the cell is opposed. Hence, the electrical gradient and concentration gradient act on the NAD+ ion. The two gradients reach an equilibrium at a membrane potential called as NAD+ membrane potential. Figure 7.33 cellular membrane with transmembrane. Proteins. The cell membrane comprises of a phospholipid bilayer which has many transmembrane proteins like channel proteins which aids in exchange of ions across the membrane. Steady state of the cell where there is equilibrium between ion pumping and ion leakage is known as resting membrane potential. The resting membrane potential of neurons is between -50 and -75 millivolts. In neurons, within the cell, potassium ions are found at a higher concentration, whereas sodium ions and calcium ions are found outside the cell at a higher concentration. Difference in the concentration of sodium ions and potassium ions provide a concentration gradient for the ions to move out of their respective channels when open. As a result, potassium ions flow out of the cells while sodium ions move into the cell. As the cell is in the resting state, it is mostly permeable to potassium ions and the resting membrane potential is influenced by these ions. When additional ion channels open, movement of ions causes a change in membrane potential, and this will be determined by the difference between resting membrane potential and equilibrium potential. Change in membrane potential imitates an electrical impulse. An action potential has three phases: depolarization, repolarization, and after hyperpolarization. As the voltage-gated channels open, the action potential begins at the axon hillock. NAD+ channels in the membrane open, but due to higher concentration of NAD+ 10 times higher than the inside of membrane, the ions rush into the cell. This influx of sodium ions is guided by concentration gradient. Movement of ions alters the voltage inside the cell as compared to outside. This phase is called the depolarization, and the resting membrane is at a voltage of -70 millivolts, which becomes less negative after entering the sodium cation inside the cell. Once the cell is depolarized, sodium ion channels start to close. Positive potential inside the cell leads to opening of voltage-gated potassium channels, which causes the potassium ions to move out. This leaving of potassium ions takes the positive charge with it, thus the membrane potential becomes more negative and begins to move back to its resting state potential. This phase is called as repolarization, and the voltage moves back to -70 millivolts. During the outward movement of potassium ions outside the cell due to the continuous opening of potassium voltage channels, the voltage shoots the resting membrane potential, and this phase is called as hyperpolarization. Every action potential is followed by refractory period, which can be divided as absolute and relative refractory period. As the sodium channels close after an action potential, they enter an inactive phase irrespective of membrane potential. This is called as absolute refractory period. Gradually the sodium channels come out of inactivation, and this phase is known as the relative refractory period. During this phase, the neuron can be excited with an external stimulus stronger than the one usually needed to initiate an action potential. Figure 7.34. Figure 7.34 graph showing the phases of action potential. The voltage MV measured across the cell membrane against time. The action potential starts with depolarization, followed by repolarization, which overshoots the resting membrane potential into hyperpolarization, and finally the membrane comes back to rest. Structural organization of central nervous system. The central nervous system comprises the brain and spinal cord. It is the control center that combines information and coordinates movement throughout the entire body. In the central nervous system, sensory neurons receive the input signals, and the signal is directed towards the motor neurons that supply muscle cells and glands. Additionally, homeostasis of the internal environment is maintained by certain association neurons that associate appropriate motor responses with sensory stimuli. The brain is a vital organ that has a protective covering called the skull or cranium. Apart from cranium, the brain is protected by cerebrospinal fluid and meninges, which are the membranes of connective tissue. Similarly, the spinal cord is also protected by the vertebral column. Meninges. Meninges are the connective tissue membranes that provide covering to the brain and spinal cord for protection and nourishment. There are 3 layers, and each layer is a meninx. The outermost layer is called as dura mater. It is a tough, white fibrous layer that is made up of dense irregular connective tissue. It attaches the brain and spinal cord to cranial bones and spinal cord, respectively. The middle layer is called as arachnoid mater, which is made up of thin network of collagen and elastic fibers that lack blood vessels. Arachnoid layer has a space underneath filled with cerebrospinal fluid called the subarachnoid space. It reabsorbs cerebrospinal fluid. The innermost layer is called a pia mater, made up of thread-like strands that contain capillaries for nourishing the brain and spinal cord. This layer forms a network of capillaries called plexuses which produce cerebrospinal fluid. This is an avascular layer and is firmly connected to the brain surface and spinal cord. Pia mater follows convolutions of gyri and sulci of the cerebral region of brain and fits into grooves and indentations. These protective meninges are in continuation with each other and surround the brain and spinal cord. Brain. Figure 7.35 parts of the human brain. The forebrain consists of structures such as thalamus, hypothalamus, and cerebrum. The midbrain and hindbrain together forms the brainstem. The midbrain consists of auditory and visual regions. The hindbrain consists of pons, medulla, and cerebellum. The brain is the processing center of the human body and functions as an integrated whole. The adult brain mainly consists of forebrain, midbrain, and hindbrain. Forebrain is the largest and in the developing brain. It is derived from prosencephalon. It consists of cerebral cortex and other small regions located at its center like thalamus, hypothalamus, and pineal gland. Midbrain is derived from mesencephalon and is the connecting link between forebrain and hindbrain. Hindbrain is derived from rhombencephalon in the developing brain. It is the lowermost, back portion of the brain, including brainstem. Medulla oblongata, pons, and midbrain are collectively known as the brainstem. Figure 7.35. Forebrain. The division of brain responsible for sensory information processing, thinking, perceiving, producing, and understanding language and controlling the motor function is the forebrain. Two major parts of the forebrain are diencephalon and telencephalon. Diencephalon is the posterior part of forebrain containing hypothalamus, thalamus, and epithalamus. It is mainly responsible for motor control functions, controlling autonomic functions, and relay of sensory information. Telencephalon is the anterior part of the forebrain and contains cerebrum, the largest part of the brain. Cerebrum is site for information processing. Hypothalamus. Hypothalamus is located superior to the brain and inferior to the thalamus. Hypothalamus has many diverse functions, such as the production of antidiuretic hormone, ADH, growth hormone releasing hormone, GHRH, and oxytocin. It also plays a role in maintenance of homeostasis by regulation of various visceral activities. The hypothalamus is also involved in memory and emotion as a part of the limbic system. Thalamus. Thalamus is located superior to hypothalamus and inferior to the cerebrum. Thalamus is mainly concerned with sensations, except for sense of smell. Sensory impulses first enter the thalamus before getting processed in the cerebrum region of the brain. The thalamus constitutes about 80% of the diencephalon, which contains two oval masses of gray matter. These areas serve as relay stations of sensory impulses. Epithalamus. Epithalamus is part of the dorsal portion of the forebrain and consists of the pineal gland, involved in the onset of puberty and the biological clock of the body. Cerebrum. Cerebrum is the largest part of the brain consisting of two hemispheres separated by longitudinal fissure. The two cerebral hemispheres are connected by attractive white fibers called as corpus callosum, which provides a pathway of communication between the two halves. The cerebral cortex is divided into lobes and each hemisphere has a frontal lobe, parietal lobe, temporal lobe, and occipital lobe. Figure 7.36. The cerebral surface of each lobe has convolutions and grooves called gyri and sulci, respectively. Gyri consist of nerve cell bodies and dendrites and is made up of gray matter. Sulci surround the gyrus. Larger, deeper sulci are called fissures. Gyri and sulci form the folded surface of the cerebral cortex, which increases the surface area, forms brain division and enables more cerebral cortex to fit into the skull. Central sulcus is a space that separates the frontal lobe from the parietal lobe, whereas the lateral sulcus separates the frontal lobe and temporal lobe. Within the frontal lobes are the motor area and premotor areas. Motor area generate impulses for voluntary movement and the premotor areas is involved with motor skills. Overall, the frontal lobe is responsible for emotional regulation, planning, reasoning and problem-solving abilities. The parietal lobe has sensory areas that receive impulses from receptors present in skin and help in the interpretation of these sensations. In addition to sensory areas, there is also taste area which has receptor in the oral cavity and the impulses are received from taste buds. The parietal lobe areas are responsible for integrating sensory information, including touch, temperature, pressure, and pain. In the temporal lobe area, the olfactory area responsible for sense of smell. Olfactory area has receptors in the nasal cavity which send impulses to the olfactory lobe for sense of smell. Temporal lobe also has auditory areas that receive impulse from inner ear for hearing. Hippocampus is a region of the temporal lobe that collects information from various parts of cerebral cortex and responsible for memory, learning, and emotions. To sum up, the temporal lobe is responsible for sensory information processing mainly for hearing, language recognition, and memory formation. The diencephalon receives the output of the impulses from the cerebrum. However, an olfactory area directly connects with the cerebrum. Figure 7.36 parts of the cerebrum. Each cerebral hemisphere is divided into four lobes: the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. Midbrain. In the developing brain, the midbrain is a small region between the hypothalamus and pons, which is derived from the mesencephalon. Midbrain, along with hindbrain, make up the brainstem which connects the forebrain and hindbrain. This region is involved in visual and auditory responses. It is also integrated with writing reflexes that help to maintain equilibrium. Midbrain encloses the cerebral aqueduct that is located dorsal to the pons and ventral to the cerebellum. Cerebral aqueduct connects the third and fourth ventricle and is surrounded by gray matter called central gray. It also forms the roof and floor of the canal as it passes through. Tectum is the dorsal part of the midbrain responsible for auditory and visual reflexes. Tegmentum is the ventral portion made up of a network of nuclei that receive and send information through the cranial nerves and is responsible for cardiovascular and respiratory controls. Hindbrain. Hindbrain is derived from rhombencephalon in the developing brain and extends from the spinal cord. It is the lowermost, back portion of the brain including brainstem. Hindbrain is composed of myelencephalon and mesencephalon. Myelencephalon consists of medulla oblongata, which performs vital functions such as controlling heart rate, breathing, and digestion. Mesencephalon consists of cerebellum and pons, which are involved in maintaining balance and equilibrium, coordination of movement, and conduction of sensory information. Pons. Pons, in Latin means bridge. It is a part of the brainstem that links medulla and thalamus. It is present on the anterior surface of brainstem attached to the cerebellum, a thick bundle of white matter, and the gray matter present underneath is continuation of the tegmentum from the midbrain. Gray matter in the pons region contains neurons that receive input in descending manner from the forebrain that is sent to the cerebellum. Medulla. Afterbrain or myelencephalon is the region of embryonic hindbrain from which the medulla originates. On the exterior of medulla, significant amount of white matter is present, which is in continuation with the white matter of the spinal cord. Midbrain, the tegmentum region and pons continue into the medulla as the gray matter is responsible for processing of cranial nerve information. In the brainstem, a diffuse network of pathway called reticular formation connects the spinal cord, cerebrum, and cerebellum. This reticular formation is related to sleep and wake and overall consciousness. The medulla contains centers that are responsible for vital functions such as regulating heart rate, blood pressure, and breathing. Cerebellum. Cerebellum is also known as the little brain. The fourth ventricle separates the cerebellum from medulla and pons and it is inferior to the occipital lobe. The cerebellum looks similar to cerebrum as it covered in gyri and sulci. The cerebellum compares information from cerebrum with the sensory feedback from the periphery through the spinal cord. Output from the cerebellum sends a descending signal to the spinal cord to correct the messages going to skeletal muscles. 6 myelinated nerve fibers called cerebellar peduncles connect the cerebellum to the brainstem and form communication pathways to parts of the central nervous system. Most functions of the cerebellum are concerned with movement such as coordination, regulation of muscle tone, and maintenance of posture and equilibrium. Brainstem. Midbrain, pons, and medulla oblongata of hindbrain are collectively known as brainstem. It is a stop-like structure that connects the brain to spinal cord. Brainstem performs basic involuntary functions such as regulation of breathing, heart rate, eating, and sleeping. Gray matter within the brainstem consists of many brainstem nuclei for sympathetic and parasympathetic functions. Out of the 12 cranial nerves, 10 arise from cranial nerve nuclei. Ventricles and cerebrospinal fluid. There are 4 interconnected, fluid-filled cavities within the brain, 2 lateral ventricles, third ventricle and fourth ventricle. Each ventricle contains a network of capillaries called choroid plexus, which forms the cerebrospinal fluid from the blood plasma. The cerebrospinal fluid is mostly water, small molecules and electrolytes. Oxygen and carbon dioxide are also dissolved in the cerebrospinal fluid and can diffuse through the different ventricles. It also provides nutrients to the neurons of central nervous system, removes waste products and acts as cushion for the central nervous system. The ventricles develop from the neural tube, the central canal. The first and second lateral ventricles are deep within the cerebrum and are connected to the third ventricle by 2 openings called the interventricular foramina. Interventricular foramina are channels that allow the cerebrospinal fluid to flow from lateral ventricles to the third ventricles. Third ventricles open into the cerebral aqueduct that passes through the midbrain and is located between the left and right sides of the diencephalon. Fourth ventricle receives the cerebral aqueduct and this ventricle is the space between the cerebellum and pons and in center of posterior fossa. Occipital lobe. Occipital lobe is the visual processing center in the brain. The visual processing center is distinguished into the primary visual cortex and secondary visual cortex. The primary visual cortex receives information from the eyes and the signal is relayed to different areas of the secondary visual processing area, which interprets depth, distance, location, and the identity of seen objects. beneath the cerebral cortex are the subcortical nuclei that enhance the cortical processes. Basal forebrain is the primary location for the production of acetylcholine, which is a modulator of the cortex activity and leads to attention to sensory stimuli. Medial lobe structures such as hippocampus and amygdala are involved in long-term memory formation and emotional responses along with the adjacent cortex. In the cerebrum there are a set of basal nuclei that are responsible for comparison of cortical processing with the general state of activity in the nervous system to influence the likelihood of movement taking place. The major structures of the basal nuclei that control movement are the caudate, putamen, and globus pallidus, which are located deep in the cerebrum. The putamen is mostly deep in the anterior regions of the frontal and parietal lobes. Together, the caudate and putamen are called the striatum. The spinal cord. Spinal cord transmits impulses from and to the brain. As the spinal cord continues its growth in the newborn, on its surface are markings of anatomical features. On the anterior side, the midline is marked by the anterior median fissure and the posterior midline is marked by the posterior median sulcus. Axons of the central nervous system enter the posterior side through the posterior nerve root, thereby marking the posterolateral sulcus on either side. Axons enter the anterior side through the ventral nerve root. Posterior regions and anterior regions have sensory functions and motor functions, respectively. During the development, the spinal cord is divided into the basal plate and alar plate. Basal plate will become the anterior face of the spinal cord and is closest to the ventral midline of the neural tube and gives rise to motor neurons. The alar plate gives rise to sensory neurons that receive input from the periphery and is located on the dorsal side of the neural tube. The base of the skull has openings called foramen magnum through which the spinal cord extends to the level of the first lumbar vertebra and continues with the medulla oblongata. The spinal cord is protected within the vertebral canal and is enclosed by meninges and cerebrospinal fluid. The length of the spinal cord is divided into regions that correspond to the regions of the vertebral column. The name of a spinal cord region corresponds to the level at which spinal nerves pass through the intervertebral foramina. The spinal cord is segmented with each segment giving rise to a pair of spinal nerves. There are 31 pairs of spinal nerves that are named as 8 cervical pairs, 12 thoracic pairs, 5 lumbar pairs, 5 sacral pairs, and 1 very small coccygeal pair. In the cross-section, the spinal cord has the shape of oval. At the distal end of the cord, many spinal nerves extend beyond the conus medullaris to form a collection that resembles a horse's tail. This is the cauda equina. The spinal cord functions as a conducting pathway for the transmission of impulses to and from the brain. The direction of transmission of impulses differs for sensory and motor impulses. Sensory impulses in the spinal cord travel on ascending track to the brain and the motor impulses travel on descending tracks. Spinal cord reflexes are involuntary response to a stimulus that do not depend directly on the brain. Reflex responses are quick and do not require conscious processing of thought. The reflex arc is a functional unit of the nervous system. For example, with the withdrawal reflex, the reflex action withdraws the affected part before you are aware of the pain. Many reflexes are mediated in the spinal cord without going to the higher brain centers. 5 parts of a reflex arc are receptors, sensory neurons, central nervous system, motor neurons, and effector. Receptor detect a change and generate impulses, and these impulses are transmitted by the sensory neurons to the central nervous system. From the central nervous system, impulses are transmitted by the motor neurons to the effector. Finally, the effector performs the designated action. Just as the brain consists of white matter and gray matter, the spinal cord also has white matter and gray matter. The gray matter is distinguished into an anterior horn, posterior horn, and lateral horn. The anterior horn is responsible for sending motor signals to the skeletal muscles. The posterior horn is involved in sensory processing. Lateral horn is a major component of the sympathetic division of the autonomic nervous system and is found in the thoracic, upper lumbar, and sacral regions. The gray matter is surrounded by an external layer of white matter. The white matter is separated into anterior columns, posterior columns, and lateral columns. Sensory information to the brain is carried by ascending tracts of nervous system fibers, whereas the motor signals from the brain are carried by the descending tracts. In the spinal cord, the columns extend as continuous bands of white matter along the length of the cord. Anterior columns are located between 2 anterior horns and are bound by axons of motor neurons, emerging from the gray matter area. The posterior columns, on the other hand, are located between the posterior horns of gray matter. Lateral columns are matter present between the posterior horn and axons of the anterior horn neuron and on either side of the spinal cord. Figure 7.37. Figure 7.37. The spinal cord. Spinal cord has a central canal filled with CSF, gray matter in a butterfly-shaped configuration, and white matter outside. Major peripheral nerves emerge. There are 31 pairs of spinal nerves named based on the emergence at the level of the spinal cord. The nerves are named as follows: C1 to C8 are 8 pairs of cervical nerves, T1 to T12 are 12 pairs of thoracic nerves, L1 to L5 are 5 pairs of lumbar nerves, S1 to S5 designated as 5 pairs of sacral nerves and 1 pair of coccygeal nerves. The numbering of nerves is done from superior position to inferior, and each nerve emerges from the vertebral column through the intervertebral foramen at its level. C1 is the first nerve that emerges between the first cervical vertebra and the occipital bone. C2, the second nerve emerges between the first cervical vertebra and the second cervical vertebra. Same for C3 to C7. However, C8 emerges between 7th cervical vertebra and the first thoracic vertebra. In case of thoracic and lumbar nerves, nerves emerge between vertebra with the same designation and the following next vertebra in the column. Sacral nerves emerge from the sacral foramina of the vertebra. Cranial nerves. Nerves that are associated with the brain are called as cranial nerves and are of 12 pairs. Cranial nerves emerge from the brain and carry impulses for the sense of smell, taste, sight, hearing, and equilibrium. Some cranial nerves also carry impulses to muscles of the face and spinal and cranial nerves. Spinal nerves. Spinal cord has spinal roots which are anchored to the dorsal and ventral aspects of the cord. Dorsal root is attached to the dorsal aspect and the ventral root is attached to the ventral aspect of the spinal cord. Each dorsal root contains sensory fibers from skin, subcutaneous tissues, deep tissues, and viscera. Whereas, the ventral roots contain motor fibers to skeletal muscle comprising of preganglionic autonomic fibers. Formation of the dorsal root is by the neuronal processes that carry afferent impulses into the spinal cord and by those processes that arise from neurons collected together to form spinal ganglion. The peripheral processes from the dorsal root ganglion neurons arise directly within the organ or structure from which they are conveying sensation. Ventral root formation takes place by processes of neurons in the gray matter of the spinal cord. Dorsal and ventral roots join to form a spinal nerve. Each spinal nerve further divides into a dorsal primary ramus and ventral primary ramus. Primary rami supply skin and muscles found on the back, whereas ventral rami supply limbs and rest of the trunk. Ventral rami intermingle to form plexuses in the cervical and lumbosacral regions from which eyes supply pharyngeal arch structures. Cranial nerves and spinal nerves differ from the other in their development in relation to special senses. Some cranial nerves have more than one ganglion and the optic nerve is attracted to the central nervous system rather than a peripheral nerve. Table 7.6. Reflex arc. The main function of spinal cord is to relay information to and from the periphery and is under the influence of the brain. Spinal cord reflexes are involuntary response to a stimulus that do not depend directly on the brain. Reflex responses are quick and do not require conscious processing of thought. Reflex arc is a functional motor neuron, which innervates the same muscle. The motor neuron, the second order neuron, is in the anterior horn of the spinal cord and causes contraction of the innervated muscle. An example of a monosynaptic reflex is the stretch reflex. Polysynaptic reflexes have multiple neurons in the reflex arc, including one sensory neuron, one motor neuron, and one cranial nerve. Major function reflex and response assessment. Eye olfactory sense of smell. 2 optic afferent pathway for vision and light. Menace response, pupillary light reflex, swinging light test. 3 oculomotor pupillary constriction extraocular muscles others. Pupillary light reflex medial movement of globe. IV trochlear extraocular muscle dorsal oblique. Ventrolateral rotation of globe. V trigeminal sensory to side of head and face. Motor to muscle of mastication. Ear, eyelid, and lip facial reflexes, pain perception from head septum chewing, jaw tone, muscle mass temporalis, masseter, pterygoid. V abducens extraocular muscle, retractor oculi, extraocular muscle. Lateral rectus. Eyeball retraction, corneal reflex, lateral movement of globe. 7 facial motor to muscles of facial expression. Ear, eyelid and lip facial tone reflexes and movement, facial symmetry. 8 vestibulocochlear afferent branch of vestibular system. Sense of hearing. Head posture, induced eyeball movement, normal vestibular nystagmus, normal gait, blindfold test response to noise. 11 glossopharyngeal vagus accessory sensory and motor to pharynx and larynx. Swallowing, palpitation, gag reflex. Nasal tube, endoscopy, slap test. Vagal and recurrent laryngeal nerves. 12 hypoglossal motor to tongue size and symmetry. Table 7.6 cranial nerves with major functions. Unit of the nervous system. Spinal reflexes are of 2 types, monosynaptic or polysynaptic. In case of monosynaptic reflexes, only 2 neurons, a sensory and a motor, are involved in the reflex arc. Sensory neuron. The first order neuron is in the spinal ganglion and gathers pulses from the muscle and sends this information to the motor neurons between them, thereby making indirect communication. An example is the withdrawal reflex. The endings of skin have sensory neuron for sensing stimuli like hot water and warm temperature. Strength of the signal is proportional to the strength of the stimulus. If the sensory endings bring about change in membrane potential that is strong enough, an action potential will be initiated at the axon adjacent to the sensory endings. The axon of the peripheral sensory neuron contacts another neuron in the gray matter by entering through the spinal cord. This contact is a synapse which results in release of chemical signal from the axon terminals due to change in membrane potential, graded potential. The chemical signal initiates an action potential at the initial segment of the neuron that transmits to the thalamus of the brain through the sensory pathway. Simultaneously, information to the next neuron is passed by another synapse. As the signal reaches the cerebral cortex, it concludes the sensory pathway. Then, the primary motor cortex of the frontal cortex sends a motor command. An action potential is sent from the upper motor neuron to the spinal cord with the target being the dendrites of the lower motor neuron in the gray matter. From the lower motor neurons, the axons emerges and connects to a muscle through a neuromuscular junction to cause contraction of the target muscle. Figure 7.38. Receptors equals heat pain receptors in skin. Relay neuron. Effector equals muscle in arm. Motor neuron. Sensory neuron. Spinal cord CNS. Message to brain. Reflex arc. Figure 7.38 reflex arc. An example is the withdrawal reflex. Synapses. Neurons that transmit impulse to another neuron do not have any physical contact or do not touch each other. A small gap or space is present between axon of one neuron and dendrites or soma, cell body, of the other neuron. This gap or space is known as synapse. At the arrival of an electrical nerve impulse, a chemical neurotransmitter is released from the terminal end of the presynaptic axon. This is a chemical synapse. Synapse can be of two types between electrically active cells, chemical synapse and electrical synapse. In electrical synapse, ions pass from one cell to the other, thereby depolarizing. Figure 7.39. An electrical impulse is generated when a chemical neurotransmitter diffuses through the synapse and combines with specific receptors present on the cell membrane of postsynaptic neuron. The axon carries the generated electrical impulse to the next synapse. The neurotransmitter is then inactivated to prevent unwanted impulse. Synapses can be either excitatory if the neurotransmitter leads to depolarization of the postsynaptic neuron or inhibitory if the neurotransmitter leads to hyperpolarization. An example of synapse is the acetylcholine neurotransmitter found in the neuromuscular junctions NMJ, central nervous system and in peripheral nervous system. Postsynaptic membrane becomes permeable to sodium ion when acetylcholine is released and this leads to depolarization. Acetylcholine is inactivated by cholinesterase, which is an inactivator. The receptor protein is a nicotinic acetylcholine receptor and sarcolemma is the postsynaptic element in the muscle cell. Neurons that secrete the neurotransmitter sometimes reabsorb the chemical themselves. This is called reuptake and leads to inactivation. Few examples of neurotransmitters include GABA, dopamine, norepinephrine, serotonin and glutamate. The synaptic end bulb has voltage-gated calcium ions channels that open when an action potential reaches the axon terminals. The outer surface of neurotransmitter vesicles has proteins that associate with the calcium ions as the concentration of calcium ions inside the end bulb in the presynaptic membrane fuses with the vesicle for the release of neurotransmitter in the space between cells known as synaptic cleft through exocytosis. Calcium ions mediate the fusion of membrane and vesicle. After release into the synaptic cleft, the neurotransmitter diffuses to reach the postsynaptic membrane. Here, receptor and neurotransmitter interact, making the binding a specific chemical event. Figure 7.39 the synapse. Transmission of impulse at a synapse with the arrow indicating the direction of electrical impulse. Neurotransmitter systems. The different systems of neurotransmitter are the cholinergic system, glutamatergic, GABAergic, glycinergic systems, serotonergic system, dopaminergic system, and adrenergic system. In the cholinergic system, acetylcholine is the neurotransmitter and there are two types of receptors, nicotinic receptor and muscarinic receptor. These two receptors are names of drugs that interact with the receptor in addition to acetylcholine. Nicotine binds to the nicotinic receptor and muscarine binds to the muscarinic receptor. Amino acid group, glutamatergic, GABAergic, and glycinergic systems belong to the amino acid group. Glutamatergic include glutamate, example serotonergic system, dopaminergic system, and adrenergic system. In serotonergic system, serotonin is derived from tryptophan. In dopaminergic system, dopamine is a derivative of tyrosine. In case of adrenergic system, epinephrine and norepinephrine are also derived from tyrosine. Neuropeptide, chains of amino acids that function to form neurotransmitter molecules. Include enkephalin, 5 amino acid molecule and beta endorphin, 31 amino acids long. Peripheral nervous system. The peripheral nervous system comprises of cranial nerves, peripheral nerves, spinal nerves, and neuromuscular junctions. The peripheral. One of the 20 naturally occurring amino acids. GABA-gamma-aminobutyric acid is also a derivative of glutamate. Glycinergic is a derivative of glycine. Biogenic amine. These neurotransmitters are synthesized from amino acids enzymatically. Nervous system comprises of nerves that connect to various parts of the body and send information in the form of signals from the brain and spinal cord. These signals are relayed by the motor nerves to the periphery for performing functions like breathing, digesting, and heart rate. Sensory nerves of the peripheral nervous system carry the information back to the brain. In the peripheral nervous system, axons run together in bundles to form fibers and fibers form the nerve which contain connective tissue and blood vessels. Nerves reach out to gland, organs, and muscles throughout the body. Based on the type of neurons carried, nerves are classified as sensory, motor, and mixed nerves. Based on direction of flow of information, afferent neuron and efferent neuron. Afferent nerves bring information to the brain, towards the central nervous system. Sensory neurons are afferent. They receive signal for hearing, vision, smell, taste, and touch and pass the signal to the CNS to encode the appropriate sensation. Afferent neurons play a role in maintaining homeostasis. Afferent nerves consist of efferent neurons that carry signals from the central nervous system to various organs and muscles to bring about an action. For example, motor neurons. Sensory organs. Introduction to sensory organs. Organs that help us to perceive the external environment and internal environment, taste, smell, hear, and touch are known as sense organs. Sense organs are equipped with receptors that are capable of receiving stimulus to convert them into an electrochemical signal. These signals then relay information to the central nervous system through sensory neurons. The 5 sense organs present in human beings are eyes, nose, ears, tongue, and skin. Eyes. Eyes receive light stimuli and provide a sense of sight and hence are visual sensory organ. Eyes can detect and focus on light image and are responsible for vision, color differentiation, and maintenance of the biological clock. Human eye can differentiate around 10 to 12 million colors. The lens of the eye can create an image of an object just as a camera is both focus, gathers and transmit the light. Most of the information about the surroundings and world around us is brought about by the visual senses. Though human eyes are considered to be round in shape, they are actually spherical and elongated horizontally. The eye is located in the eye socket of the facial skull with only 1/6 portion of the eye visible in the front. The eye consists of 3 layers: outer, middle, and inner. Fibrous tunic is the outer layer, vascular tunic is the middle layer, and neural tunic is the inner layer. Figure 7.40. Outer fibrous layer. The outer layer is made up of sclera and cornea. Sclera is a tough protective fibrous layer that shields the inner structure of the eye and has white appearance. The optic nerve passes through the sclera at the back via an opening and forms majority of the fibrous layer. Sclera transforms into cornea as it reaches the corneal limbus while descending to the anterior portion of the eye. Cornea has a dome-shaped curve that helps focus light entering the eye on the retina present at the back of the eye. Cornea is a transparent wall that covers the iris and the pupil. Cornea can regenerate itself after a corneal injury or abrasion because of the stratified squamous epithelial cells present at the border that constantly split and regenerate. Middle vascular tunic. The middle vascular layer is also known as uvea and includes the iris, pupil, choroid, and ciliary body. Iris contain the melanin pigment which imparts color to the eyes. Intensity of eye color is proportional to the concentration of melanin. Dark brown eye color indicates high melanin, whereas blue eye color means low level of melanin. Behind the cornea, iris is present and is composed of circular muscle called sphincter pupillae and radial muscle called dilator pupillae. Pupil is the opening of the iris present at the center and the size of pupil is controlled by sphincter pupillae muscle and dilator pupillae muscle. Pupil size changes with light intensity. When the light is bright, the pupil size reduces because the sphincter pupillae muscle tightens. In dark, the opening of pupil increases in diameter because the dilator pupillae muscle pulls the iris radially or outwardly from the pupil to allow more light to enter the eye. After entering the cornea and pupillary opening of iris, the light comes in contact with the lens which is big index transparent structure. Lens is located in the posterior region of the eye chamber and due to its big index nature can alter its shape to let the light enter the eye. The lens can become flattered or rounder and this change in shape is controlled by a structure called ciliary body which includes ciliary muscle and projections called ciliary processes. Lens is held together in position and is able to change shape because of the ciliary processes that connect to suspensory ligaments, and the ligaments are directly attached to the lens. Lens becomes flattered when the ciliary processes pull on the suspensory ligaments due to relaxation of ciliary muscles. Choroid muscle is dark brown and can absorb light. It is also full of blood vessels to provide nourishment to the eye. Without choroid, light would not be focused on the retina and is most likely to be scattered and hence visual processing is interrupted. Inner neural layer. Inner tunic is the inner layer and has the retina. Retina consists of ganglion cells that synapse with bipolar cells which further synapse with rods and cones, the photoreceptors. Retina is the pigmented region made up of a single thick layer of pigmented epithelial cells. Light entering the eyes first reaches retina, then goes past the ganglion cells and bipolar cells to finally hit the rods and cones. Light that does not hit the photoreceptors is absorbed by the pigmented layer to avoid scattering of light and preventing from bouncing back to photoreceptors. Rods and cones. Rods and cones are the photoreceptors present in the eye. Rods are highly sensitive to light and can be activated by a single photon. Due to their sensitive characteristic, they can be used in low light conditions. However, the vision perceived is only black or white. Each eye contains approximately 120 million rods. Cones are less sensitive to light in comparison to rods and can detect either red, blue, or green wavelength of light. On seeing. Figure 7.40 structure of the eye. The sphere of the eye can be divided into anterior chamber and posterior chamber. The walls of the eyes are divided into three layers: the outer fibrous tunic, the middle vascular tunic, and inner neural tunic. Within the neural tunic lies the retina. A red object, only the red cones are activated, whereas, on perceiving purple object, both red cones and blue cones are activated. Each eye contains 6 million cones. Rods and cones act as transducers by converting electromagnetic energy to electrochemical energy. Light, once enters the eyes as electromagnetic energy, hits the rods and cones, thereby activating them to generate an action potential. Action potential generated is electrochemical energy. This conversion of electromagnetic energy to electrochemical energy is done by rods and cones, and then the optic nerve sends a signal to the central nervous system and brain to process and visualize. Chambers of the eye. The eye as a whole is divided into anterior chamber, posterior chamber, and vitreous chamber. The anterior chamber is the region from cornea to iris and is filled with a transparent liquid called aqueous humor that is secreted by the ciliary epithelium. This fluid provides nutrients to the lens and cornea and also provides structural shape to maintain the shape. Posterior chamber is the space between iris and lens and is filled with another liquid called vitreous humor. Vitreous chamber is the space between lens and back portion of the eye. Ears. Human ears convert mechanical energy into nerve impulses, which is then transmitted into the brain, hence acting as transducers. Anatomically, ear can be classified into three parts, that is, external ear, middle ear, and inner ear or internal ear. Figure 7.41. Transmission of sound to the internal ear is done by the outer ear and middle ear portion. Internal ear is for hearing and maintains the balance system of the body. Outer ear. Outer ear is the first, visible part of the ear that consists of pinna and the ear canal. Pinna, also called auricle, at the bottom has a fleshy lobe known as earlobe or lobule. The overall shape and size of the ear is due to the pinna, which is made up of cartilage. The sound waves are directed to the ear canal by the pinna. Ear canal, also known as external acoustic meatus, is a short curved tube that starts at the temporal bone and ends at the tympanic membrane and is about 24 millimeters in length. The tympanic membrane, also known as eardrum, is a thin translucent membrane. Ear canal contains hair follicles and beneath the skin is the ceruminous gland. Thick, sticky yellowish earwax is secreted by the ceruminous gland, and the function of earwax is to prevent foreign objects from entering the tympanic membrane. The shape of the tympanic membrane is conical and it separates the outer ear from the middle ear. Sound waves touch the tympanic membrane and are then transmitted to the middle ear. Middle ear. Middle ear is a small region that is housing three smaller bones or ossicles. The three bones are named after their resemblance to a hammer, an anvil and stirrups malleus, incus, and stapes, respectively. Stapes is the smallest bone in the entire human body. Within the temporal bone, the middle ear is present as an air-filled cavity with the shape of a chamber containing a floor, a roof, and four walls. On the later side of the middle ear, eardrums are located, and on the opposite side lies the internal wall, also called medial wall. The middle ear is separated from the inner ear by means of the internal wall and has an opening above, the oval window, and an opening below, the round window. The mastoid cavity is located within the temporal bone with the help of mastoid antrum, and the middle ear is connected to this cavity by the posterior wall. The anterior wall of the middle layer is connected through eustachian tube to the nasopharynx. The eustachian tube equalizes pressure on the tympanic membrane, prevents reflux of fluid from reaching the middle ear, and cleans the secretions of the middle ear. The epitympanic recess is the roof of the middle region of ear. Inner ear. The inner ear is also known as labyrinth. It consists of cochlea, vestibule, and semicircular canals. Cochlea is attached to the vestibule at the front and semicircular canals towards the back, and the vestibule behaves as a pathway. The semicircular canals help with balance and equilibrium. The inner ear is made up of bony labyrinth which form the tough outer layer, and on the inside is the membranous labyrinth, and both of these are filled with fluids, perilymph and endolymph, respectively. Perilymph and endolymph flow are necessary for hearing and balance. The cochlea has the shape of a snail's shell and converts sound waves into electrical impulses. In a cross-section view, cochlea contains three parts: scala vestibuli, cochlear duct, and scala tympani. Scala vestibuli contains perilymph and is connected to the middle ear through the oval window. Cochlear duct is filled with endolymph and houses several organs of Corti. This is the main region of hearing and contains hair cells that transduce wave motion into the neural signal. Scala tympani contains perilymph and is connected to the middle ear through the round window. Figure 7.42. Figure 7.41 structures of the ear. The external ear contains the auricle, ear canal, and tympanic membrane. The middle ear contains the ossicles and is connected to the pharynx by the eustachian tube. The inner ear contains the cochlea and vestibule, which are responsible for audition and equilibrium, respectively. Figure 7.42 inner ear structure. The three major spaces within the cochlea are highlighted. The scala tympani and scala vestibuli lie on either side of the cochlear duct. Physiology of hearing. Sound vibrations start to travel from the external ear and then reach the ossicles present in the middle ear. Sound waves are directed towards the eardrum by the ear canal in the presence of air, and the canal causes the waves to vibrate back and forth. The central area of the eardrum is connected to the middle ear, to the malleus ossicle which starts vibrating. From malleus, the vibration is transmitted to the other two bones, first to incus and then to stapes. Stapes, the middle ear ossicle passes on the vibration to the inner ear. Stapes has a back and forth movement which leads to pushing of the oval window inward and outward, thereby causing vibration. As the oval window vibrates, fluid pressure waves in the perilymph of cochlea is set up. Pressure waves are transmitted from the scala vestibuli to scala tympani of the inner ear and then to the round window. Pressure waves in the perilymph also causes back and forth movement of the vestibular membrane, thereby creating pressure waves in the endolymph of cochlea. Pressure waves along with the endolymph pressure wave causes basilar membrane to vibrate. This results in movement of hair cells against the tectorial membrane. As the hair cells move, a signal is sent to the brain, represented by a movement. As the nerve impulse reaches to the brain, different sounds can be distinguished. Equilibrium. Along with hearing, the ears also associate the equilibrium. Equilibrium, the sense of balance is maintained by the inner ear. Hair cells present in the inner ear act as mechanoreceptors and have stereocilia which are mechanosensitive organelles. These hair cells in the semicircular canal can detect rotational or dynamic equilibrium such as head movement. Another set of mechanoreceptors present in the utricle and saccule of the vestibule recognize static or gravitational balance such as head positions. Dynamic equilibrium. The vestibular consists of three semicircular canals in utricle and saccule. In the semicircular canal, a membranous semicircular duct is present which contains endolymph and open in the utricle. At the end of semicircular canal there is an enlarged portion that detects changes in head rotation. This enlarged portion is called ampulla and change is detected by its dynamic equilibrium. Ampulla has hair cell, which are balance receptors. When the hair cell detects movement of endolymph fluid inside the canals due to movement of the head, an electrical signal is sent. This signal reaches to the brain when it is picked up the vestibular branch of the eighth cranial nerve. Static equilibrium. The macular region which is present in utricle and saccule contains sensory cells called balance receptors. Utricle and saccule are also filled with endolymph. When there is change in head position with respect to horizontal or vertical acceleration, it is known as static equilibrium and this is detected by sensory receptors. Static equilibrium includes inertia felt on sudden stopping a car or when inside an elevator. The gastrointestinal system. Introduction to the digestive system. The food we eat is not in suitable form to be used up by the cells of the body. The digestive system converts complex food in the form of carbohydrates, proteins, and fats into simple organic and inorganic molecules to be absorbed by the blood and transported into various locations. The remaining supply of nutrients is transported to the excretory system for elimination through anus. General organization of gastrointestinal tract and its function. The fundamental anatomical parts of the gastrointestinal tract, GI tract, and its accessory organs are illustrated in figure 7.43. The digestive system is distinguished into two parts: alimentary canal or digestive canal and accessory organs. The alimentary canal starts from the mouth and ends at the anus. The accessory organs are organs and structures that aid in the digestive process. The alimentary canal and its organs are discussed below. Digestion takes place in the oral cavity, mouth, and is assisted with salivary glands and tonsils for chewing, mastication, swallowing, ingestion, and taste. Pharynx present in the throat region helps in swallowing of the chewed food, also called bolus. Esophagus functions to move food from mouth to esophagus, propulsion, protects the food against stomach acid, and produces mucus for lubrication. From esophagus, food reaches stomach which aids in digestion, storage, and absorption of nutrients. In stomach, food is mixed to form chyme and is also protected from stomach acids. Figure 7.43 The overview of the digestive system. Large intestine is also involved in absorption, storage, mixing, and propulsion. Anus eliminates indigestible remains. Small intestine helps in digestion, neutralization of stomach acid, mixing, propulsion towards large intestine and excretion of bile juice. The accessory organs of small intestine are gallbladder, pancreas, and liver. The digestive system starts with mouth and ends at the anus and in between includes all the organs such as esophagus, liver, stomach, pancreas, intestine necessary for digestion. Intake of food, chewing, and salivary secretion. Mouth. The food enters the digestive tract from the mouth which is made up of cartilaginous skeleton. The oral cavity contains lips anteriorly. In the posterior region is the fauces. On the side is the cheeks. Upwards inside the mouth is the palate and below is the muscular floor. The buccal cavity has stratified squamous epithelium covering that protects against abrasion and it has two domains, vestibule and oral cavity. Tongue. The tongue is inserted into the hyoid bone and the back part is attached whereas the tip anterior portion is free and connected to the floor of the mouth by the frenulum present behind the lower incisor teeth. The frenulum is a thin fold of mucous membrane. The tongue is made up of voluntary skeletal muscle which are of two types, intrinsic muscles and extrinsic muscles. Intrinsic muscles play a role while drinking and swallowing by altering the shape of the tongue. Extrinsic muscles help in retracting and protruding of tongue. The anterior portion of the tongue is almost 2/3 root and posterior portion is 1/3. The anterior portion is separated from the posterior portion by a groove called the terminal sulcus. The anterior part has majority of the taste buds and has a layer of papillae, whereas the posterior portion is devoid of papillae, but consists of small glands and lymphoid tissue and lingual tonsils. The tongue helps with mastication and propelling the food into the esophagus. Teeth. Teeth are arranged in two arches, maxillary dental arch and mandibular dental arch with one arch on each jaw. The 16 pairs of teeth are arranged in 4 quadrants, upper right, and lower left. In each quadrant there is one central and one lateral incisor, one canine, 2 premolars, and 3 molars. An individual tooth has 3 parts, crown, neck, and root. Figure 7.44. In the center of tooth there is a pulp cavity through which blood vessels and nerves pass. Root canal is the pulp cavity in the root portion of the tooth. The crown portion is surrounded by enamel which helps against acid and abrasion. The teeth of course helps in chewing of food. Figure 7.44 Structure of tooth. The tooth consists of a crown, neck and root as shown in the figure above. Mastication. Teeth help in chewing the food. This process is also known as mastication. In mastication large complex food particles is broken down into small bolus that can be easily digested. Incisors and canines are the front teeth that are usually employed for cutting and tearing of food. Molars and premolars are used for crushing and grinding of food and these are present at the back portion of the oral cavity. The intensity of chewing food is controlled by the cerebrum and the reflex is controlled by medulla oblongata. Salivary glands. Saliva is the digestive secretion of the oral cavity and is produced by 3 pairs of salivary glands, which are parotid, the submandibular, and the sublingual glands. Figure 7.45. Parotid gland is just in front and below the ears. Submandibular are at the posterior corner of mandibles and sublingual is below the floor of the mouth. Saliva aids in secretion and helps to keep the mouth moist. Figure 7.45 Salivary glands. The major salivary glands, the submandibular glands, the sublingual glands and parotid gland. Parotid glands, they are the most massive salivary glands among the 3 and produce watery-like fluid without mucus called serous secretion. They are located at the angle of the jaw. The ducts of parotid ends to the front margin, open in the mouth opposite the second upper molars on each side. Parotid gland accounts for 25% of the total saliva output. Submandibular glands, they are at the posterior corner of mandibles and secrete higher amount of serous and comparatively less mucus. They contain acinar cells that secrete mucoproteins, as well as cells producing serous fluid. Thus, their secretion is more viscid than the parotid glands. They account for 70% of the daily output of saliva. Sublingual glands, these are paired glands majorly consisting of mucus alveoli and are smallest among all 3. These are present below the tongue and 10 to 12 small ducts exist and opens into floor of the membrane. They contribute to the remaining 5% of the salivary output and gives a sticky character to the saliva. Mechanism of salivary secretion. The saliva present in mouth facilities swallowing by lubricating the food and aids in speech. The saliva contains enzyme called alpha amylase or tylen for digestion of starch. As starch is digested, it secretes maltose and isomaltose which provide a sweet taste. Chewing digests about 2% minus 5% of carbohydrates in the buccal cavity. Saliva contains a variety of enzymes like lysozyme and immunoglobulin that provide protection against bacteria. During secretion glands are provided with a lubricating quality because of mucin. Per day 1,500 milliliters of saliva is produced. Saliva secretion is enhanced during hunger, smell of food or even thoughts of food. Reflexes of the salivary glands are stimulated by the parasympathetic and sympathetic nervous systems. Pharynx. Pharynx is located in the throat region and has 3 portions, nasopharynx, oropharynx and laryngopharynx. Nasopharynx consists of ciliated pseudostratified columnar epithelium. Oropharynx and laryngopharynx are covered with squamous epithelium and aids in transmitting of food. Oropharynx communicates with nasopharynx. Esophagus is a part of the alimentary canal and carries food to the pharynx and stomach. It is about 25 centimeters in length. It starts at the esophageal hiatus, opening of stomach and ends at the stomach. The walls of esophagus consist of submucosa, mucosa, muscularis, and adventitia tunicus. The muscularis has an outer skeletal muscle and an inner smooth muscle. The movement of food within the throat is regulated by sphincter and the time taken depends on the consistency of food. Swallow deglutition. The process of swallowing is known as deglutition and has 3 phases. Voluntary phase. In this phase, the food is chewed and converted into bolus with the help of teeth and tongue. Then the bolus is moved down to the throat, the oropharynx region. This is a voluntary process and takes place consciously. Pharyngeal phase. In this phase, food is moved down to the esophagus from the pharynx. As the soft palate is elevated, it leads to closing of passage between nasopharynx and oropharynx. This process takes place unconsciously and phase lasts for approximately 2 seconds. Esophageal phase. Food moves from the pharynx through the esophagus to reach the stomach. Esophagus generates peristaltic waves for the movement. This phase lasts for about 7 to 8 seconds. The stomach. Stomach is located in the upper left part of the abdomen and the size and shape vary with individuals, time, food intake, etc. Anatomy of stomach. Stomach is part of the alimentary tube, but it is a sac that extends from the esophagus to the small intestine. The tube traveling from the esophagus into the stomach is the gastroesophageal or cardiac aperture. The area surrounding is called cardiac region. The parts of the stomach are as follows. Fundus, the body is the largest part and pyloric region is the portion joining the small intestine. The opening between the two is the pyloric opening, surrounded by the pyloric sphincter. Secretions of the stomach. Stomach secretes fluids like hydrochloric acid, HCl, mucus, gastrin, and histamine which mixes with the ingested food to form chyme, a semi-fluid material. The mucus cells present on the surface secretes a 0.15 cm thick viscous alkaline mucus layer on the epithelial cells of the stomach wall. The thick layer of mucus lubricates and defends the epithelial cells of the stomach wall from the damaging impact of the acidic chyme and pepsin. Upon irritation, the lining of stomach secretes large amounts of mucus. Glycoprotein is an important element that binds with vitamin B12. This results in easy absorption of B12 in the ileum. Pepsinogen is secreted by gastric chief cells and is converted to pepsin by HCl and pepsin molecules present inside the lumen. The parietal cells in the gastric glands near the pyloric region secrete intrinsic factors and concentrated HCl. HCl is responsible for creating a low pH environment, 1,3 pH. It functions majorly to kill bacteria as well as holds digestive properties on ingested food. Low pH inactivates salivary amylase that can hinder carbohydrate digestion. Figure 7.47 illustrates the formation of HCl in parietal cells. Figure 7.46 Anatomy of stomach. A. Internal anatomy of stomach showing all the sections of it. B. Showing types of cells present. Figure 7.47 HCl production in stomach. 1 carbon dioxide, CO2, moves across the cell from the bloodstream. 2-Ca2 combines with water H2O to form carbonic acid H2Ca3 under the influence of carbonic anhydrase Ca. 3H2Ca3 dissociates into hydrogen ion H+ and bicarbonate ion HCO3. 4HCO3 travels back into the bloodstream while chloride ion Cl countertransport enters into the cell. 5H+ travels into the duct of the gastric gland. 6C enter diffuse with the charge H+ within the gastric gland to form HCl. 7 some potassium ions K+ are countertransported into the cell in exchange for the hydrogen ions. Regulation of the stomach. Stomach produces nearly 2,500 milliliters of gastric juice every day and 700 milliliters is secreted due to food. Regulation of these secretion is controlled by hormonal and nerve mechanisms. Gastric secretion is also controlled by chemical signals like gastrin, secretin, gastric inhibitory polypeptide, and cholecystokinin, as well as the paracrine chemical signal histamine. The three phases of stomach regulation are cephalic, gastric, and intestinal phase. Cephalic phase. In this phase, gastric secretions of acid and pepsinogen starts at the thought, sight, taste, smell, and by food in mouth. These perceptions stimulate the medulla oblongata containing centers within the vagal nerve. Parasympathetic stimulation in the stomach cells by the postganglionic neuron results in release of acetylcholine ACH. Acetylcholine results in release of gastrin, which in turn stimulates the chief cells and parietal cells to release hydrochloric acid and pepsinogen. When acetylcholine, gastrin, and histamine work together, flow of hydrochloric acid is higher. Gastric phase. This phase occurs when the food has reached the stomach and the maximum amount of secretion occurs. Swelling of the stomach wall in presence of peptides due to ingestion of food leads to stimulation of gastric secretion. This leads to activation of mechanoreceptors that stimulate the intrinsic nerve plexuses to secrete gastrin, HCl, mucus, and pepsinogen. Gastric secretion stops when the pH of the stomach is in Lt2, thus the amount of gastric juice produced is reduced due to negative feedback mechanism. Intestinal phase. Food that enters the mouth is converted to bolus and then into chyme as it enters the small intestine. However, the gastric secretion is inhibited when the pH of the chyme is below 2. The acidic nature of chyme results in release of secretin which acts on parietal cells, chief cells as well as local enteric reflex which in turn inhibits gastric secretion. Furthermore, gastric secretion is also inhibited by release of cholecystokinin and gastric inhibitory polypeptide due to the presence of lipids and fatty acids in small intestine. Expansion of intestinal wall. Presence of irritants in the duodenum, low pH, hypertonic or hypotonic solution contribute to inhibition of gastric secretion as the factors stimulate enterogastric reflex. Enterogastric reflexes reduce the amount of gastric secretion. The exocrine functions of pancreas. Anatomy of the human pancreas. The pancreas function as an endocrine and exocrine gland.